O-Level Biology 5090, Paper 2, May-June 2024, question number 4 to question number 6, variant 21. So first of all, question number 4, and in question number 4, you can see two diagrams. So what is in question number 4? Some plants can survive extreme dehydration for a long time in a dormant state. They start growing again when water is available. They are adapted to survive in deserts. Selaginella lepidophylla is an example of this type of plant. So this is a desert plant, Selaginella lepidophylla. So the figure 4.1 shows Selaginella lepidophylla plant after several months without water and the same plant 3 days after rainfall. So several months without water, it shows that plant is a brown color. It means it has protected itself from adverse environmental condition, which is dehydration. So for its survival, it has protected itself in this way. But when there is rainfall, so 3 days after rainfall, you can see the leaves are there, which are of green color. So these leaves has increased the surface area for photosynthesis. So the green color shows that the chlorophyll is there in this plant. Chlorophyll is there in the plant, and due to you can say the chlorophyll captures the sunlight. Process of photosynthesis will take place. So as a result of photosynthesis, there will be formation of glucose or carbohydrate. So this glucose will be stored in the form of starch in different parts of this plant. So this starch will be used in adverse environmental condition to live alive. So this question was of 4 marks. Now I would like to move towards the next question. So next question you can see on screen is a diagram of a cell taken from this plant. So shows the cell wall and nucleus of a cell from plant after several months without water when there were the conditions of dehydration. So suggest and explain the appearance of other parts of the cell after several months without water. You may draw one on figure 4.2 help your answer. So I would like to draw on the picture as well after writing. So what will happen in this plant when there is dehydration? It means the water outside the plant is in lesser water potential as comparison to the cell. So what will happen? The water will move outside from the cell, loss of water from cell, by osmosis due to the water potential difference. So due to loss of water, the cytoplasm will shrink and then cell sap or vacuole will shrink and cell membrane will pull away from cell. So if we draw all these conditions, we can see that. So in this way, we can draw the picture when there is loss of water, and then all different and the plant in this condition is called as plasmolysed cell. Now we will move towards the next question. And the next question is, Selaginella lepidophylla is the scientific name for the plant species, which also has the common name resurrection moss. Explain what is meant by the term species. Species is the group of organisms living at a place and interbreed with one another to produce a fertile offspring. So fertile offsprings are produced. It means they belong to the same species. Next question, state the name of the scientific system used to name the species and suggest one advantage of using the scientific name rather than the common name. So binomial. So binomial nomenclature is the method of classification in which the organisms are classified into groups and into subgroups by using the two names, that is the genus name and species name. So the binomial nomenclature is the name which used to these names, that the genus and species name, to represent the scientific name of an organism. So the advantage of these names is that they the same organisms which is called by different names in different parts of the world, internationally they are recognized by single name. So we can write the advantage. Common with the same name in all parts of the world. So internationally they are accepted, or internationally they are recognized by the same name. So this is the advantage for by using the scientific name for different organisms. Otherwise, the common names, you can say the common names are different in all parts of the country. Now, the question number 5, Bacillus subtilis is a species of bacterium that is widely used in biotechnology. It can be used to manufacture the protein lipase for industry use. Is a diagram of B. subtilis. Two of the parts labeled A, B, C, and D in figure 5.1. So in figure 5.1, there are involved in the manufacture of proteins, and those two names and explain their role in protein manufacture. So those can be the A and D. First of all, what is A? Letter A represents the DNA. DNA is a genetic material, and that genetic material is, you can say, it has genes on it. So genes are used to manufacture the protein. And then the D is ribosomes. So ribosomes add amino acids together to build protein. So briefly, I would like to say that the DNA, which is copied to make the new protein, and then ribosomes, they are used to manufacture that protein, which is coded by the DNA. Now the next question is, suggest why B subtype has flagella. Flagella is a locomotive organ. So it is a locomotive organ with the help of which bacterium moves. Explain why plasmids are useful in biotechnology. So the plasmids are the circular DNAs which are present in the bacterium, and these uh, plasmodiums can be used the can be used to insert the foreign DNA, or the genes can be inserted into the plasmids. Into the plasmids. So in biotechnology, the plasmids are very helpful, as we can insert the required genes into the plasmids, and these plasmids, again, we can keep them back into the bacterium where they multiply, and in this way we can manufacture the products of our desire. Next question, part B. You can see a graph has been shown. B subtilis can be grown. Subtilis, Bacterium subtilis can be grown industrially in large containers. When B subtilis is an ideal condition, it can reproduce rapidly, increasing the population size and the biomass of bacteria in the container. Graph E and F, two graphs are shown over here. Figure 5.2 show how the biomass of B subtilis increases after small populations of bacteria are added to two containers with different environmental conditions. Now, move to the question. Give the name of the large industrial containers used to grow bacteria. So large containers which are made up of steel, and they are used to manufacture the different products on commercial scale. They are called as fermenters. In graph E, the biomass increases as shown in figure 5.2. Indicate using the letter M the part of the graph E where bacteria are reproducing most rapidly. So first of all, where the bacteria are producing more rapidly, you can see the point on the graph where the bacteria are producing more rapidly. Here you can say this is the point. So this is the point where the bacteria are start growing rapidly in this way. So we will put the M over this point. Now the next question. And in next question, you can see that the calculate the percentage increases in biomass for graph E in the first 6 hours space for working. So we have to find out, move towards the graph to find out the answer. So you can see that at point where the growth has been on graph E. So you can see it is the point. So this is the point where it has started the growth, and then you can see this is 0.4. So it was started at 0.1. So we will move towards the space to write its answer. So in this way, we have to find out its average. So 0.41 multiplied by 100, then the answer will be 400%. So this is the answer. After 6 hours, actually, will be the growth percentage. Next. Next question number 4 is, Describe two differences between graph E and graph F. So between graph E and graph F, we have to find out, or we have to move towards the graph E and F. You can see in graph E and graph F, there is increase, and graph E has higher final biomass, you can say, and F has lower biomass. So F has at lower place, you can see. So it in this way we can, so it shows the higher biomass and it can show the lower. So it's quicker, its movement is quicker, you can say. It has shown the faster movement as comparison to the graph E. So these are the two differences which can be written on the given space, and we are moving towards that space to write this answer after observing it from the graph. So first, the greater increase in biomass in at E, and secondly, the rate of increase is also fast. Faster in E. Next question, suggest one environmental condition which could have caused the differences between graph E and graph F. So graph E and graph E, that can be, when there is, you can say, growth takes place inside the fermenter. So in that conditions, we have to keep some environmental conditions constant, because they can cause the problems in the production of those products. So they should be, the pH should be maintained, the temperature should be maintained. So they need to maintain all these different conditions. So move to the next question. Lipase is also produced by the human body. Explain why it is produced and how its function is different from the function of bile. So lipase digest is used, lipase is used to digest the lipids, whereas bile has no enzyme. No enzyme. So bile emulsify the fats. Emulsify the fats. The larger molecules are broken down into small droplets to increase their surface area. So in this way, both are different from each other. Bile lipase is an enzyme, whereas bile don't have any enzyme. So the lipase will break down or digest the lipids or fats into fatty acids and glycerol. So on the other hand, the bile, which don't have any enzyme, but it emulsify the fats into small droplets. Now, question number 6, and question number 6 is, a beam of bright light shines into a human eye for 2 seconds. A similar beam of light shines onto one side of the tip of a plant stem for several days. Describe the ways in which the human and the plant coordinate their responses to their beam of light. Your answer should include, first of all, stimulus detection and the method of responding. So first of all, we will discuss about human. As you know, the human in human body, the eye receives the light. So receptor cells are present. Cells are present on the retina. And then a message is sent towards central nervous system, and then iris helps to control the entry of light into eye by reducing the size of pupil, because in this way the eye is prevented by damage. On the other hand, what happens with the plants? You know, the plants are they will detect the light will be detected by shoot tips, auxins, which are growth hormones. They move move towards shaded side and increase its length on that side. Length of that side. So the other side will show movement towards light by bending. So in this way, the stem will show the positive phototropism towards the light. Now the next question, we are moving towards the next question, and next question is, describe the role of synapse in a reflex arc. So what are the, first of all, what are the synapse? Synapse is a gap between two neurons. So it is gap between two neurons, so it will help in the transmission of nerve impulses between, helps in the transmission of nerve impulse from between two neurons. You can say that between two neurons. Unidirectional, it also help in one way direction of nerve impulse. And secondly, it also help in the transmission of in the movement of neurotransmitter from one neuron to the other by binding from one. So in this case, what happens at the time when there is the movement of the neurotransmitter from one neuron to the other, you can say in this way, the movement takes place and the neurotransmitters are in the form of chemical messenger. So from one neuron to the other neuron, they are diffused with this cell membrane of the one neuron and they are released into the synaptic cleft. So in or the base of the other neuron, there are the receptors for these messengers. So they will attach themselves with those messengers. So in this way, they will move from one neuron to the other neuron. So it was all about the part 2. Please like, share and subscribe my channel, and thanks for watching.